A scientist measured the rate of uptake of a monoglyceride and a monosaccharide by epithelial cells of the small intestine of mice. A monoglyceride is a molecule of glycerol with one fatty acid attached. She did this for different concentrations of monoglyceride and monosaccharide. So we can see the straight line here is for the monoglyceride, and the curve that's levelling off is for the monosaccharide. And question 6.2 asks us to use knowledge of transport across membranes to explain the shape of the curve in figure 1 for uptake of monosaccharides between concentrations. So looking back at the curve, we can see that it starts to increase as the external concentration of the molecule increases, so does the rate of uptake by epithelial cells, but then we get to a stage where it will level off. So from the shape of that graph, we can conclude that the movement across the membrane is due to facilitated diffusion, and it is through a carrier protein. Between A and B, we can see that as the external concentration of the molecule increases, then so does the rate of uptake by the epithelial cells. So they are proportional to each other. As one increases, the other one also increases. But then when we get to part C and D, we get the levelling off, because at this point here, the rate of uptake of the epithelial cells remains constant, because facilitated diffusion is using channel and carrier proteins, and at this point all of those proteins must be saturated. They must all be in use, so your proteins in the membrane are your limiting factor. So 1 mark for a reference to facilitated diffusion, or the idea that carrier proteins are involved in the movement. They will also accept channel proteins. And mark 2 for a description between A and B, that the rate of uptake is proportional to the concentration, or a description of proportional. And between C and D, then all the channel or carrier proteins are in use or saturated or limiting. They are the limiting factor.